The broadcast is now starting. All attendees are in listen-only mode.
Good afternoon and welcome everyone to our Calf Health Refresher webinar today. Thank you so much for joining, and thank you to Catherine McAloon, who has kindly offered to participate or to present this webinar. So I'm just trying to move my slides and of course they won't work. So hopefully you'll all know Catherine anyway from the vet hospital, but she's also the chairperson of the CalfCare TWD, which AHI run. Also, a very special thanks to Jenny O'Rourke there in the background and for planning this event. We really would be lost without her, and Natasha Mounier, another colleague in AHI, for helping out. So just a few housekeeping. The webinar is being recorded, so mics are muted, but if you do have questions, we encourage you to type them in. You'll see the type in question box there on your dashboard at the side. I just want to also remind you of our AHI events calendar. That's on the AHI website, and that that can keep you up to date with all our future training and events that are coming up. And also, we are involved in a new research project between Teagasc, DAFM, ECD and AHI, with Pablo from Moorpark is leading it, but it's trying to develop a tool for farmers in reducing their antimicrobial use. But he is looking for advisors or vets or farmers that will be interested in participating in this, even just to kind of get advice on what we should be doing from the start. So there is a QR code there. If any of you are interested in participating in that, we'd encourage you to register your interest on that link. So just the department asked us to update you all on a recent update from them in relation to Schmallenberg. Sorry, there's a QR code after coming up there for some reason, but so it's just to let you know that they have had a lot of evidence of circulating Schmallenberg in late 2024, so that there are likely to be an increase in cases in calves and lambs this spring. So just to make you aware of that, and that based on past experiences, that these outbreaks will be unevenly distributed and probably more common in synchronized calving or lambing if they were affected at a particular time. So just to be more vigilant, try and encourage your clients to be more vigilant. And if they do find or come across any abnormalities, they really would encourage you to get them submitted. Just when blue tongue is circulating in Europe, we can't take it for granted that it is the deformity is or the abortion or poor calf is from Schmallenberg. So just for extra vigilance and caution around that, it would be great if you may, don't want to frighten every farmer, but just to be aware that there is an increase. So I'll hand you, just one more thing. So we don't do many of these webinars, but if it's something you'd like to see more of, or in general, what you would like to see more of from us, we have put together a SurveyMonkey, just 6 questions that we would really appreciate if you could complete it after this webinar. Jenny will also send a link by email to anyone who has registered. So without further delay, I will hand over to Catherine. And again, thanks so much, Catherine. We'll take questions. I'll probably try and keep until the end, unless Catherine feels like a break going through her presentation. But would encourage you to type them in if you have any. The more engagement we get, we'll all learn from it. So hopefully I've stopped sharing, Catherine, and you'll be able to start sharing. Yeah, brilliant.
Can you see my screen and hear me okay, Michelle?
You can. It's not in slideshow though, it's just like
yeah. Should be us now, is that right? Yep.
Yeah. Thanks.
Apologies myself, and so firstly thank you for the invitation, and we were asked kind of as CalfCare group to do this more training for vets and advisors, so I'm aware that there's a mix in the audience. I have done very similar for the kind of grad search and in the past, so I doubt there will be anything terribly new, but I suppose the point really is that it's a refresher given the time of year. So apologies, I've had my share of the January doses, so my voice hopefully will hold up. And also myself and GoTo don't have the best relationship, so hopefully we don't hit any technical difficulties. What I am going to talk about really is just a quick whistle-stop review of some of the ongoing research that we're involved in on calf health, and then a quick kind of summary on the calf health in terms of BRD, bovine respiratory disease, and calf scour. Talk a little bit about performance metrics, and then we'll talk a little bit about diagnostics, BRD and housing assessment, and kind of ideas to approach these on a herd level problem, both for respiratory and disease and calf scour. So I suppose it's always important to set the scene. So we know, last year there was 1.5 million dairy calves born, or more than that actually, in to a dairy breed dam in Ireland. I suppose the biggest thing we have is that greater than 1 million of those calves are born in the first 11 weeks of the year. So that can be a very difficult concept when we're talking about comparing our calf rearing systems with our competitors, particularly in Europe and so on, that this real concentrated calving brings such a pressure on calf rearing. So I guess we all know health and welfare are key to part of any farm system or any successful farm system, and calves are always part of the kind of welfare focus. So many dairy heifer calves that are born will alive at a month of age do not actually make it to their third lactation, and a certain percentage, 10 to 15%, we estimate, don't actually make it to first calving at all. So that obviously has a huge implication in terms of efficiency, both globally and at the farm level. And it is one of the most significant costs on a dairy system. That is the cost of getting a heifer calf to the parlour and when they start to pay you back. So we're talking in the rounds of €1,500. I would imagine that estimate is gone up if we were to redo some of those figures just now. I suppose it's also important that it is not just about having calves alive as they enter the production cycle, whether that is as a dairy beef animal or a dairy animal. But this idea is very well it's increasingly well understood as time goes on. So it's not just about kind of managing them to keep them alive, and this is kind of where the collision between a labour efficient and maybe very economical efficient system maybe doesn't quite meet the kind of health welfare side. So I suppose really what I mean here is that it's been shown that improved average daily gain, pre-weaning average daily gain, actually can have a much longer term downstream effect on that animal when they hit the parlour 2 years later. And it's about they're born with a certain amount of genetic potential, but how we manage them pre-weaning and that early life environment has been shown to influence how the expression of that potential. So I suppose moving on after that, then just to talk about some of the key kind of research things that we're involved in. I guess some of you will have possibly heard of this before in terms of that we did a pilot study. It's 2023 now. Where does it's 2 years old now? Where does time go? But I guess summer scour was something that's kind of been really topical over the last few years. First grazing season calves close to turnout or relatively recently weaned. There is a school of thought that this appears later in the summer as well. But that these essentially first grazing season calves that develop a really intractable scour, ill thrift with ulcers, potentially oral ulceration, and there's a huge spectrum of severity within the cohort affected. So you might have a few of the kind of strong calves mildly scouring right through to calves that are so dehydrated they can't stand up. So this is something that we've investigated kind of several outbreaks over the last few years, and I guess when we went to talk about this around the country, it seems to be a huge problem for the farms that suffer it. And it is not by any means the level of a BRD or scour or a calf scour problem that every farm will run into, and it's also not consistent year on year. So hypotheses as to where summer scour came from, and even the name of it is debated in an international audience, but the hypothesis is related to exposure of these young calves to lush grass, and whether that is to do with high protein or potentially an ammonia issue in the lush grass, or potentially a ruminant acidosis, or it is related to how they are weaned, whether that is not complete or successful weaning. I suppose I would add there is other suggested causes and they are probably different conditions that are standalone in their own right, the likes of molybdenosis or other potential mineral issues, undetermined viruses, for example. But ultimately, the one common agreement is that the calves are all exposed to grass. So in 2023, we ran this pilot study between UCD and Teagasc. So Rishi is the PhD student, perhaps some of you have come across. And we asked vets to refer these suspect cases for an investigation. And I suppose, interestingly, lots of them were ruled out at the first screening stage. So they were on the fecal egg counting that was done initially. Worms and coccidiosis was reported. And so I guess lots of farms looked like the summer scour issue, I suppose, was kind of the key point, and often were not. We had less than anticipated number reported that year. If you remember, there was hot weather suggested to be potentially protective, and that can be a controversial, but certainly there was less than expected. So the farms that we did go to, we looked at calves, examined them, took samples of blood, rumen fluid, scanned their lungs, looked at a detailed history of feeding, took grass samples, etc. So these are the kind of calves, I'm sure you're well used to seeing them on your travels, these kind of, again, a spectrum. Some are housed and they're shocking looking, lovely calves going out and they fall apart. And I would also, I suppose, point out one of the key things we did come across was this, was the kind of grass management of young calves seems to vary wildly. So some reported strip grazing. I'm by no means a grassland expert, but I think we would certainly question the agreement if that was truly kind of the point of strip grazing, because they were on such a large allowance, etc. And again, this idea that comes up year on year, and it's this kind of varying of severity within the cohort affected on farm. Interestingly, a few of the farms, and it'd be something that is sort of separate to summer scour that we would see through UCD quite a lot, is that there was other things going on some of these farms. So, they presented as a very typical presentation of summer scour, but in actual fact were diagnosed with, a percentage or a cohort of them having respiratory disease, and some having molybdenosis. So you'll see there a very high mgs per kg molybdenum on the fresh forage that the calves were in. And in the absence of a very robust animal level test, kind of grass molybdenum is one thing that we do look at quite a lot. So I suppose the point really here is that some of this syndrome, as it's called, the summer scour, we're talking about cases that are, have molybdenosis ruled out, have BRD ruled out, and where there's no other kind of plausible explanation. So I suppose it does present very similar to other conditions of recently weaned and grazing calves. So you may have heard me talk about this before, or some of my colleagues involved in this project. The take-home message really that we did find from this pilot study was that on the case farm, so case farm 1, 2, 3 there, that's across a range of number of calves sampled per farm. The blood ammonia, so measured as a point of care test, that has to be done calf side. Ammonia is so volatile that really, unless even if it's frozen and taken to the lab immediately, it tends to escape and can be, falsely elevated otherwise. So they talk about trying to test it as a point of care measure. So we were running these samples calf side on farm with a handheld monitor. And the take-home message really was that we find very high levels of blood ammonia in these affected calves, and similar calves in the farms, we call them the non-case farms, that turned out to have an outbreak of BRD or molybdenum. We actually had a few other control farms that were investigated for mineral issues that aren't shown here, and they all had ammonia concentrations well within the normal range. So this was a very interesting finding, as it supports the original hypothesis that maybe a high presentation of very high protein grass to these developing ruminants is a potential reason that might explain some of the issues seen. I'll come back to that in a moment. So this was a very small study, pilot study. High blood ammonia and the point of care test is used in pretty much most other species. It's used in dogs, it's used in horses, and it has been used and published in calves before. And it's very interesting that the affected, or at least the worst affected calves, all affected calves on affected farms showed the same common finding. We did measure their rumen pH, and we took other measures of rumen function that were subsequently analysed later, lactate and other VFAs and so on. And they were certainly not ruminal acidosis, and with a high blood ammonia, they were on the opposite side of the spectrum. The weaning in those calves seemed adequate. That was based on kind of what the farmers told us. We did think it was interesting that lots of fertilizer was used in calf grazing and also applied quite close to when the calves were turned out. And as I mentioned already, kind of sort of wild variation of interpretation on what is strip grazing. So, ultimately calves were out on large paddocks that may have had a wire, but certainly were not forced to eat stem, which we would have potentially said was, it was defeating the purpose really. And that we know calves are browsers and will just eat the leaf in preference. So they were well able to do that even in farms where strip grazing was being supposedly applied. I guess what was really interesting, and I think is something that is an important concept, is it is very difficult to accurately measure dry matter intake in this kind of cohort of animal. So on the back of that, we thought that looking at fertilizer and grassland management was something that would be useful. So in 2024, a study in Teagasc Grange was undertaken to look at really robust, healthy calves turned out to, on the first grazing season, to a different rotation of low, medium and high nitrogen fertilizer applications to the grass. Additional findings are, and I suppose the take home is that we did not manage to induce it. That is summer scar in these calves. So I suppose it's been, the analysis is ongoing and it's not published yet, but I think it's interesting because one, our hypothesis could be completely wrong. Or two, I think what the real difference of these calves, it was that there was no issue around weaning. So kind of in control of all sourced from one farm, and the calves were all doing really well at turnout and so on. And I think it does sort of highlight the issue that this may be a combination of, or at least the kind of working hypothesis is that this is more likely a combination of a failure of weaning or less than appropriate weaning in combination in the face of a high protein challenge in the grass or high nitrogen. So look, that's the study that is being analysed and written up at the minute, and it does, I think, probably raise more questions than answers. So it's certainly not going to be solved straight away. But one thing I suppose we have come out, and I guess it's fairly well, practiced now, is that ultimately if we see these farms that we should be trying to rule out coccidiosis and worms, as they often present very similar, and that actually examining a cohort of the calves and making sure that, it's not like a chronic BRD type issue. The grass sample cut with scissors to avoid soil contamination and sent for mineral analysis is an absolute must, really, to rule out the likes of a molybdenosis that would be managed differently. And really it's about triaging the kind of mobs of calves, housing the worst of them, grouping accordingly, maybe reducing group sizes, kind of minding the worstly affected ones. But ultimately it comes down to diluting that grass. So anything we can do to dilute the exposure, which we know is first grazing season turnout or relatively lush grass. So whether that is strip grazing, offering an alternative forage outside, putting them in, doing anything to try and increase their fibre intake and dilute the kind of grass substrate. So I suppose anecdotally it seems to work very well for most farms. I know there is the odd exception. So it does either, and a lot of farms now are moving to the kind of doing this preventatively or at least reactively. So it does reduce the number that are kind of very thoroughly investigated. Just to briefly touch on some of the other kind of key areas on calf research, I suppose some of John Donlan's work I think is relevant, I suppose, at this time of the year. And that, I suppose, is that in keeping with other international research, there is no one ideal calf house, but that high airborne ammonia, high particulate matter, or kind of particulate matter, dust and so on, high airspeed or drafts and wet bedding have been shown to increase the risk and need to be avoided in terms of control of BRD. The prevalence that he found at herd level, a 4% prevalence of respiratory disease, but very, very variable within herd prevalence. So I suppose we know that we can have farms with huge amounts of their calves suffering from respiratory disease, but that the next door neighbour perhaps might have very little issues. Other work that's ongoing that we're involved in at the moment is looking at the colostrum source in the development of the calf microbiome and its impact on diarrhea development and feeding of waste milk to calves. So that study done by Anna Flynn in Moore Park, showed quite predictably that calves that were fed antibiotic contaminated milk under Irish conditions went on to develop both phenotypic and genotypic antimicrobial resistance. They did, however, perform similarly, and I think that's interesting because it does kind of tempt us into saying, oh, well, maybe it'll be okay, but it does raise the question about what we do with waste milk. And I guess it does show that very predictably feeding antibiotic contaminated milk to calves does very much alters their microbiome and this development of resistance genes. The second study completed by Anna that's in review there at the moment, I think will be interesting, once it's out in the ether, is that we looked at, well, the team in Moorpark looked at the impact on growth rate by feeding different amounts of milk replacers. So a total feed rate of about 15% of birth weight was fed, but they were fed in different allowances at a varying max allowance of 6 litres, 7 litres, or 8 litres. And I suppose what was interesting is that the calves that were stepped down at 4 weeks of age did fall back compared to those that were delayed to kind of later at the kind of 6 weeks. So again, kind of supporting the idea that we should be sort of pushing them out to that 6 weeks before we consider them as a transitional ruminant. So just a kind of snapshot of what's going on in Ireland in terms of the calf research sphere at the moment. So moving on then to the calf health, I suppose what we're supposed to be here to discuss. I guess it goes without saying, and we know from the passive surveillance reported by DAFM every year, that the leading cause of death in neonatal calves under a month of age is calf scour, and from 1 to 6 months of age is respiratory disease. Rotavirus and cryptosporidium are our leading causes of the etiological agents involved in calf diarrhea. And I guess, that there is the very typical kind of age susceptibility, that, as we're all probably well aware, the kind of 1 to 4 weeks of age is that kind of really susceptible time. I suppose, interestingly, cryptosporidium in combination with rotavirus as a mixed infection are the most common causes of calf scour across the globe, basically, and that has been kind of repeatedly reported. I think what's interesting about crypto, and just as a quick reminder without going too heavy into the pathophysiology of it, is that it is a parasite, and it is with a double-walled outer shell. So it makes it incredibly resistant to environmental contamination. It just hangs around there and remains infective for a very long time. It has a very low infectious dose. So, ultimately in a situation where we have 1 or 2 scouring calves, that infection pressure in the shed, in the pen, goes off the charts very quickly because they only need a small number of oocysts in a healthy calf to induce infection. Once they become infected, they shed in huge numbers, further perpetuating the problem. And I suppose very importantly to remember that it is zoonotic. So I guess in my job, every year we have veterinary students that get cryptosporidium, whether it's from their farm experience or their clinical placements. So I suppose it's worth always reminding our clients, because that's the calves are notorious for those who have never been exposed, or immunocompromised individuals, for potentially setting it up as a human infection. And I suppose as a note, it is notifiable. So if a human goes to the doctor and it's confirmed as cryptosporidium, it does invoke a HSE investigation. So I suppose moving on from the kind of other etiological agents, you could talk about them all day. There's lots yet to be identified. This kind of, as whole genome sequencing becomes more readily available and fairly advanced now, lots more bugs are being identified in calf scour, but their role as causal or kind of incidental finding is a lot harder to determine. And really, we're still kind of confident that the main players are the ones that were used to the cryptos and the rotas. So the same old problems, really. In terms of the BRD spectrum, I guess this is another one. I think it can't really be overstated. We see this time and time again, this idea of the environment, the bacteria, the virus, the immune system, the management, and how they all interplay. But the more I study respiratory disease in calves, the more I realize the environment is actually the kind of you can't avoid it. I suppose it can't be disentangled from the pathogen story, and ultimately the modifiable risk factors. Of course, we've got things like vaccination and so on. But a lot of respiratory disease management factors do tend to fall to kind of managing environmental risks. And we've also completely changed our approach to diagnostics for respiratory disease in recent years, with the movement towards thoracic ultrasound now being very common. So I guess just to kind of the main ones we know, bovine RSV, PI3 would be the 2 key ones in young calves, with a potential role of bovine herpes or IBR in some cases. And then, with anything you like yourself, in terms of if you looked for it, you would probably find coronavirus, influenza D, bovine adenovirus, etc. More commonly now, with kind of advent and access to these of the molecular testing, these things tend to be found. But when they are studied as standalone pathogens, they tend to be rarely virulent enough to cause an outbreak of disease. But what they do is similar to the RSVs and the PI3s of this world, the coronavirus and influenza D and so on, can do the initial insult that makes that calf much more susceptible to the subsequent bacterial invasion, your mycoplasma, your pastorellas, your manhinias. Histophilus, and they are the ones, I suppose, that once they are given any path, that they will set up a raging and horrible pneumonia. There's some really interesting work done in Liverpool, and actually we're looking at human models of disease, and then we're looking at how air pollution and particulate matter affected respiratory cells, and they've also looked at some of this in bovines now across the world in different research groups, and we've shown that very similar to other species, this idea of the mucociliary escalator, so the little tiny hairs that beat in the trachea that serves as the natural immune defense for the respiratory system, is very heavily impacted by the initial viral infection that subsequently allows the setup of the secondary bacterial pneumonia to reach the lower respiratory tract. But in addition to viral insults, things like cold stress, so other stressful events and draft have also been studied as things that can directly impact the kind of role of the immune function in calves. So I think that's where the interesting interface with the environment piece comes in. So in terms of looking at calf health problems at the group level, which is something that I'm sure most of the audience here will be faced with over the coming months, I guess firstly we can look at calf health as part of the review of any herd health production cycle on any farm, dairy beef, dairy suckler, etc. You know, are they reaching their performance targets in terms of their weaning weights, their mortality, morbidity, etc. Not treating too many, and there'll be different targets depending on the system, obviously. Or it can be an actual subset of the herd health cycle where there's a specific problem investigation, which tends to be the more urgent and emergent in the spring, where the wheels have fallen off and, you know, an outbreak scenario has occurred, and it's about really trying to get in time and stop that or control it where possible. So what are the key production indices for calf rearing? And I think this, I get a little, I suppose, uncomfortable with trying to give a specific one answer to this. And for the reason for that, I would say, is that for the farms that we would go to, for example, you try and quantify performance at what it is first. So we try and look at that as the kind of first step. And, you know, if they're running up to treating 30% of calves, or it would not be uncommon in an outbreak scenario to have mortality hitting 15-20%. That's mortality now, not morbidity. In a full outbreak scenario, we have gone to pneumonia cases where there's 30% mortality, same with a crypto outbreak, with other maybe complicating factors. So it would be highly unrealistic for that farm to be trying to get to a 3% mortality in you know, a few weeks or a year or whatever. They need to be, it's a sort of a moving the goalposts year on year and a gradual process. Equally well, if the farm is doing quite well and wants to do better, then, you know, obviously best practice, we'd like to be hitting mortality certainly less than 3% with an alarm threshold of 5%. So that is between birth and weaning I'd be talking about there. And morbidity. So morbidity is not quite the same as treatment, but ultimately we kind of use treatment records as a proxy for this. So pre-weaning scar or respiratory disease, we certainly wouldn't like to be treating more than 10% of the group, or we would say there's a problem, possibly raising that to maybe 15% of the group if you're looking at the post-weaning period. But again, it really does tend to be farm specific. You know, a farm that's running at 30% treatment is unlikely to get to less than 10 in 1 year. So I guess it really sort of should be kind of tailored a little bit to the farm and the expectations. Other KPIs, I suppose, and a lot of them do revolve around growth rate, and that's kind of hard to avoid because, you know, an acceptable growth rate and acceptable average daily gain, if measured correctly, usually is a proxy measure for how well the calves are doing. So, you know, by inference, they tend to have better immune function, they're not cold stressed, and you tend to be seeing less issues. But I guess the original literature on average daily gain in pre-weaned calves would have come from indoor kind of intensive systems with accelerated milk feeding, you know, where they would easily be doing 0.8, 0.9 kilos a day. And the reality is that will certainly not be realistic, based on different genetics and on different feed systems. That, you know, they're not going to do that on 15% of birth weight on a 21% powder or something like that. And, you know, we'll have lots of farms here where we'll have an average birth weight, maybe in heifer calves born to heifers of 30 kilos. That wouldn't be that uncommon. You know, and we ask them to double their birth weight in 8 weeks or 56 days. You know, they really only need to be doing 0.6 to get there. So I guess, again, it needs to be kind of bespoke to the farm. There is evidence, Kira Hayes' work, in which he was in UCD, showed a positive effect on reducing days open at the subsequent breeding season for maiden heifers of about 0.8 to 0.9 average daily gain in the pre-weaning period. she also showed a positive effect in terms of first lactation milk solids based on calves that grew better in that pre-weaning period. So again, that's back to that epigenetic kind of side where we try and turn on their genetic potential as much as we can with how we manage them. So I guess, but equally well, we are very rarely looking at accelerated growth programs, milk feeding very high litres in our kind of seasonal production system. So there's no reason farms can't do that if they wish. But I guess having an average daily gain that is appropriate to the system, like maybe 0.6 is perfectly fine for what they're feeding and what they want to do. I suppose is the point, rather than taking 0.8. And again, it will depend whether we're there to investigate a problem or whether we're looking at, just sort of setting up a general what they need to do. And again, in that case, we would need to look at breed and work backwards from the kind of target pre-breeding weights and so on. So in terms of an outbreak scenario, we can do lots of nice graphs and things like that by looking at mortality and morbidity rates in Excel. We can do attack rates, epidemic curves, all that kind of interesting things. But ultimately, we really just need to look at treatment records and decide, the number of calves at risk and how many were treated, and are we okay with that or not, or is this a sign that we really need to intervene or change? We do look for patterns. I think that would be important regardless of the amount of data. So is it calves that are 1 week old, or is it calves that are 6 weeks old, or is it really the very much closer to weaning? I'm trying to really hone in or distill the kind of the pattern in time or space. And ICBF, herd software and paper records will be used to do that. So I guess from our point of view, looking at calf deaths or calf health issues, we will try and split it by male, female, by breed, by age, by month calved, and so on, just the kind of things you can do. But very simply, as a look-see and a starting point, and a very robust starting point, would be to look at the mortality records based off the stock reconciliation report on ICBF. And that's a kind of a good starting point, and maybe that's as much as you need to do to sort of start and catalyse the conversation about calf health. So on farm then, I think it is important everybody's on the same page. What is the farmer's perspective? But within that, I would say, I think what's really important in terms of investigating a calf outbreak is that it's narrowed down. So it's, when we go to the farm, I find that, particularly from students or anything like that, it's very easy. It's just natural and conversation to kind of, well, this calf died last week, and then there was another one died of meningitis, and there might have been another one died of navel. And I guess it's not about every calf death, it's about the kind of pattern. So most of the calves are dying with diarrhea that develops between 2 and 4 weeks of age, and that the mind is focused on looking at that as opposed to maybe every potential issue, because I think it's difficult then in order to sort of make meaningful kind of targeted changes. And then of course we want to know how many are affected, age at onset, whether they're getting better in response to treatment or not, etc. Understanding the farm system kind of goes without saying, as a lot of these would be your client, whether you're an advisor or a vet. But I do think it's incredibly important when it comes to calf rearing. So certainly the bigger farms, some will have night calvers, for example. Maybe they'll have night calvers for the first half of the season and not the second. Maybe the calves then will have a completely different colostrum protocol depending on staff as the season progresses. Maybe it's a smaller family farm and different people feed the calves every day. Those kind of things do become relevant in an outbreak scenario. So I guess that's what I mean by kind of really understanding the detail of the system. Then we try to deal with the actual, deal with the actual details. So how many calving pens are there, how long do the calves spend in there, how clean are they, when are they cleaned out, what is the exact protocol that happens to a newborn calf, and the real detail there. So when I say what is the colostrum management, that is not that they get 3 litres quickly, it's how exactly is it harvested, is it in a dump line, is it hand milked, do they go to the parlour, how soon after calved are they when it's collected, how is that collection vessel managed, where is the colostrum stored, that kind of detail. And then obviously the plan of nutrition, which I'll come back to, becomes really important. So what are they fed, for how much, for how long? Water quality, I think incredibly overlooked in calf rearing. We did 2 visits kind of very close together. I remember just around Paddy's Day last year, and both farms were not offering water to the calves till the calves were about 3 weeks of age. Which I kind of blew my mind, but I guess, offering water, how soon is it put in, is it clean, that kind of thing. And I suppose maybe more for things like abomasal bloat and so on, that will become really important. But ultimately, water for calves is something that can't be forgotten or overlooked in terms of the history taken. The hygiene, that's quite difficult to do crude assessments of, but ultimately we do want to be looking at the milk feeding equipment, the milk bars, the buckets, probably cutting open a few teats. Potentially, we're also talking about the hygiene of the calving pen, the calf accommodation. So that's quite a very broad term. Understanding the structure of the calf dynamics. So are they housed with older calves? What are the group sizes? What is the kind of age range? Are the calves all within a week or two of age, or are they, maybe 4 weeks of age? Housing assessment, I'll come back to that. And then husbandry is important. So when are they actually disbudded? When are they vaccinated? Are those things done at the same time? Again, it sounds common sense, but in the face of an outbreak, it's amazing what might come out in the wash, and how weaning is managed. So I guess after the kind of history gathering, we want to actually see, whether that will happen at the same time. Ideally, we would really look at calf feeding, for example, examine a cohort of calves, and plan our diagnostics accordingly. And I think that's where I come back to this diagram, and not to overstate it or be too repetitive, but it's easy. I know when I'm talking to vet students as well, ultimately our immediate thought, and it's human nature, will go to the pathogen. What's causing this? What's the virus? What's the bacteria? What's the parasite? But I suppose really, and as you'll see kind of in a moment, I hope, is that the actual animal kind of host immunity, the environment, and those modifiable environmental risk factors are probably as important to measure as the actual kind of pathogen hunting diagnostic side of things. So obviously we will look for, the sick animals. We need to look at, what age they are, how sick they are, what's the, for things like scour, for example, how severe are they? Is it degree of acidosis, all of that kind of thing. But then other animal level tests will include blood tests for passive transfer of immunity, fecal tests to try and determine the actual cause if it's a scour issue, pneumonia diagnostics, and potentially measuring the calves for weight, for growth rate. And then we'll have other tests such as the assessment of housing, assessment of current stressors, and assessment of nutrition. So I think I at least personally find it very important to look at those things as a diagnostic, so as not to distract solely from the kind of pathogen, which the pathogen testing is of course important, but not the only part. So in terms of assessing colostrum, now I would certainly be an advocate for most, realistically in this day and age, we know every other research finding on calves is related to colostrum. So it is difficult to justify and not looking at the colostrum quality. Even on the farms where things are going well, is there marginal gains to be had? The use of a Brix refractometer, and maybe they don't want to check every calf at the point of feeding the colostrum, but having a handle of the Brix readings for colostrum quality is something that is, the first port of call when you run into a calf issue, but probably and realistically should be done on all farms at this stage. In addition to Brix readings, in an outbreak scenario, usually we will take some colostrum samples and get them sent for microanalysis for TBCs, total bacterial counts, and coliform counts, just to look at how cleanly they're collected and stored. That is, I guess I would say, not a routine, but something that can be useful to really focus the mind on colostrum if that is kind of where the biggest risk factors are. So I suppose important to remember that you can have a farmer hand you, if they have recorded their Brix readings, and they might have 50 odd readings, and a lot of them are reading above 22% on the Brix, and your blood tests for passive transfer don't add up, and there looks like failure of passive transfer. That is not that uncommon. So I guess that's where we start to look at things like how clean it's collected, and how it's stored, if it's actually refrigerated or not, because those are the kind of things then that, even though it looks like it has enough antibodies, the calf has not actually absorbed them. So there's a lot in the colostrum quality piece to be investigated. Then the second half of the story on colostrum is actually taking blood samples from the calves. And I would suggest that all farms should be doing a check, kind of 2-3 weeks in, and maybe again later in the season, blood testing 10 to 12 calves, or some other sort of sensible number. And certainly I know a lot of the big units that I've been involved with would be doing this now as routine, and lots of vets are, and there's calf point of care tests and so on for this now as well. Excuse me. There's a lot of research work goes on around the testing for passive transfer of immunity, but I think a lot of it does become academic. Of course, there are pros and cons, but I guess to summarize it, I certainly would usually check for total proteins in calves aged 1 to 7 days of age, ideally. You can do ZSTs either. And we would use a cutoff. So historically, we used to use this arbitrary cutoff that total proteins of greater than 55 grams per litre in enough calves of the screen of the sample size would suggest that colostrum management and passive transfer is fine. A lot of research now, and particularly the more recent stuff, is suggesting that 55 is actually quite low, and the calves do perform better with higher levels of passive immunity. Now, there's an ongoing experiment in Moore Park that will be published shortly that has shown that kind of whether it was 22% on the bricks or 28 mightn't have made that much difference. And all calves, as long as it was good quality, did well, if it was absorbed correctly, etc. But I guess, for practical purposes, we're looking at screening a sensible number of calves and remembering that this isn't an individual animal test, which is why we don't get bogged down in well, ZST has this slight advantage or total protein has this one, and it's this pro and this con, and it just has to be practical. So screening the total protein of a group of calves, and ideally we would look at the number then that are kind of good, excellent, and fair. That's kind of, I suppose, where the kind of global direction of travel and assessment of passive transfer is looking at it, not on this kind of binary yes or no, but on kind of like a scale. So animal level tests for scour, I guess I would suggest that the blood test for passive transfer of immunity in a screen of calves is the first port of call, examining the sick calves, of course. And then, of course, we have to take feces samples to see what the pathogens are. So it is time and time again, I don't think you'll probably never say it enough times, looking at the scour will never tell you what it is. So it has to be tested. The SNAP tests and point of care tests are very useful. I suppose they give you a rapid answer. I would suggest they don't test for everything. And if it doesn't kind of fit the bill, the confirmatory testing may be required. We have run into that issue before. And of course, post-mortem samples can be useful as well, and making sure to follow up and see what was there. So it is important to know if infections are mixed, because obviously mortality tends to be higher if there's zoonosis involved, for the like salmonella or crypto. So it is important to know what is there, and also for the ancillary treatments that might be required. So there's probably no point in putting in, Halakir or Parafor or something like that to a herd where it's not crypto and it's a simple rotavirus issue. So I guess knowing what it is, I'm not suggesting every calf, but a subset are sent so that you kind of know what the pathogen is. It is important. And then the non-animal tests, so I've mentioned checking colostrum quality at the very minimal on the Brix refractometer, understanding how calves are managed when they're sick. So often mortality is higher if the calf management protocol, if the milk is withheld, fluids are not going to put in quickly enough, that kind of thing. Hygiene assessment, which I'll come back to, or housing assessment, and plane of nutrition. So plane of nutrition, regardless of whether it's scour or pneumonia, is a key element, because calves that aren't growing or are not being fed enough are stressed calves, so they need milk feed for growth, for heat, and for immune function. So if they're only getting enough for maintenance, those calves tend to get sick. And I suppose, so we're talking about, assessing the actual consistency of the routine. Are they fed at the same time every day, at the same milk temperature? Is it the same person? That kind of thing. Obviously, how much? So I would usually talk about percentage of birth weight as to, what they started on. Is that, historically we would have talked about 15%, and that's kind of the industry standard currently. The direction of travel would be much higher than that now internationally, which can be bringing a whole debate around how many times you can deliver feeds and so on. But ultimately, the Canadians, the New Zealanders, the UK would all be sort of moving towards advising 20% of birth weight as a, fed at a minimum of 2 feeds for those early weeks. And but I guess it comes down to how much, how often, is it enough? Is it being mixed up correctly? We may actually need to test the milk replacer and see how is it being weighed out. All of those kind of the detail in that may require bringing in, the advisors to the farm, the milk replacer, the automatic feeder people, if necessary, etc. So assessing the hygiene of the feeding equipment is important. So, and then also within that, then, is there access to enough clean water? Is there an issue with water pressure or hard water maybe for feeders? Are they being offered starter? What's the quality of it? Etc. So really nutrition becomes really important for immunity. So when I talk about assessing the plane of nutrition, it's really to see, is that a risk factor for why calves are getting sick? So this was one of the boards that was provided there at the CalfCare events, I suppose, just kind of summarising some of those key points for the farmer audience. Moving on to respiratory disease then, diagnostics, I suppose, again, very similar. It's going to have to involve blood tests for assessment of passive transfer, clinical examinations of the calves, then the pathogen tests, so that may include lung washes or transtracheal washes or a deep nasopharyngeal swab, or it might include bloods for kind of serological screen, but only when those calves are old enough, so probably not when they're out of maternal antibody cover, so probably not practical for the early spring. Postmortems are useful, but only if the correct case is sent in, so not the calf that's been sick for 2 weeks with pus at its nose, but kind of the slightly more acute or less chronic cases. We do use things like the Wisconsin calf score, so although it is biased towards upper respiratory disease because it looks at nasal discharge and eye discharge and ear position and so on, in the States, for example, that would be widely used by calf rearers to look at pen prevalence and kind of monitor interventions and so on. I don't think that's practical here, but just I suppose it is a tool that can be used. And thoracic ultrasound is becoming increasingly used here, because there's so much evidence for it being the better diagnostic. And then similar to the scar situation, we need to understand the colostrum management, the plane of nutrition, hygiene, and the assessment of housing. So to talk about BRD diagnostics, I think it is important when we're deciding on what test we're going to do. Are we looking to diagnose pneumonia, in which case it's examining calves and scanning lungs and so on, or is it actually for the pathogens involved? Is the cases suitable for testing? So have they actually not, are they recently pyrexic, for example, and they haven't been treated with antibiotics or something like that? Can we actually interpret it, and what will it add? Because BALs and transtracheal washes are not straightforward in young calves, and they do help us look for the kind of active pathogens in the lower airways, but the trade-off needs to be, will it add enough to the recommendations? So the nasopharyngeal swab is fine as long as it's a guarded one that goes right to the kind of nasopharynx, but in very acute and pyrexic cases, and being careful that, if we see Histophilus on it, okay, that's important, but some of these can live in the nose, but ultimately if they're there in the nose and there's a pneumonia outbreak, they are also likely to tell us what's going on in the lower airways. BALs and transtracheal washes are shown to be very useful, especially now we can actually interpret based on the cytology, percentage neutrophils and so on, as opposed to just PCRing for viruses and so on. Serological blood testing, so maybe you look back and say, we had a terrible spring there, and now we need to check what is circulating. Taking a sample of bloods for serology for something like Mycoplasma bovis, you can prove that it's circulating. That might be where you might employ that. Post-mortem obviously is a very useful test, again, if they are the right cases. So thoracic ultrasound is used at the calf and herd level. It is, there's really no gold standard diagnostic for pneumonia, so I'm afraid, as controversial as it might be, the stethoscope is not the gold standard either. So thoracic ultrasound, with or without the Wisconsin calf scoring, have been used very widely now in terms of research on respiratory disease. But thoracic ultrasound is also widely used in terms of both at the individual animal to see are they likely to get better, as well as for prevalence estimates and so on. So I think it's something to look at upskilling in if necessary. Just to mention the Wisconsin calf score, as I said, it's not a practical to be used on every farm, but it can be used in an investigation if need be. Just to quickly mention the idea here of thoracic ultrasound. So, lungs, aerated lungs tend to give us a nice picture with the rectal ultrasound, those that have consolidation and so on. So this becomes sort of, you can scan large numbers of calves relatively easily, relatively quickly. So I suppose it's something that you can upskill in reasonably quickly with kind of some basic training, I suppose. So something to consider. So to talk just briefly now about housing before I'm conscious of time here, the non-animal level test. So it is important to assess housing. I guess it's better to understand that is there some risk factors that we can look at. Is it the group size, the inlet, the outlet? Is there draft? Is the shed maximising the wind effect, but without draft? Is there drainage? And so on. So I suppose it is important to think about why we have calves in calf housing and BRD, because there ultimately is no ideal calf house. So we want to provide shelter, we want to provide adequate ventilation and temperature control and reduce moisture. So as I mentioned briefly already, draft, cold stress, high stocking rate, all of those things can directly impact the calf's natural respiratory immune function in the trachea, and therefore their ability to not succumb to pneumonia. And the assessment of housing for a dairy breed calf versus a suckler calf on a cow is entirely different. So, I suppose bearing that in mind as well. And ultimately, we build calf houses so that we can fit in a machine to clean them. So, but they are usually large vats of air in concrete and steel, built with concrete and steel, and are cold. So, the wind effect here is just where on most days in Ireland we have some level of wind, and where it blows across the shed. Ideally, if the shed is situated at the 90 degrees to the prevailing wind, but it works best. But what is the prevailing wind direction? Of course, as recent weather events will tell us, that is difficult to predict. But ultimately, they will ventilate on their long axis, where wind will blow across the shed above animal height, and it will remove kind of airborne pathogens, particulate matter, dust, ammonia, and fresh air will follow. Versus the stack effect, so where animals generate heat and thermal energy created from the animals basically rises to the highest point and escapes through the roof. And as that does that, it draws in fresh air at the eaves. However, the reality is that unless that is a cow-calf operation, or animals that are maybe over 200 kilos, or beef calves, that the stack effect simply doesn't work in large group calf housing. And what we have is this: it rises so much, but actually the steel air just recirculates within the shed. So in reality, most natural ventilation in Ireland relies on this, the wind effect. And it's quite rare actually that the stack effect ever really works, in at least for dairy breed or small calves. But ventilation is important, as I mentioned there, to remove the airborne pathogen, try and provide some kind of uniform distribution of air change, but equally well without causing a draft. So that's not that easy to achieve. I guess the simple things, even though we are not designing sheds for the stack effect to work, we do want to look at the inlet-outlet area for the types of sheds, because that does help us understand the wind effect, if it's working, and the kind of air change that is potential within the shed. And we also want to look for drafts. We want to look for moisture. Is there leaky drinkers, that kind of thing? Is there no drainage in the bed so that they're lying on a damp floor? Because obviously moisture facilitates longer pathogen survival and it reduces heat. So, the biggest thing for young calves is lying on a wet bed, because they're going to lose heat to the floor and they become cold stress calves. So assessment of moisture is important. Temperature is important, and look, there's so much data now that shows that the temperature inside a calf house is often colder than that outside, or at least the same. And unless there is a big temperature differential inside versus out, you don't have the stack effect. It's simply impossible. So, the lower critical temperature for calves under 3 weeks is quite high. We're talking about 10, 15 degrees, and it's usually not that in Ireland in spring. Why is temperature important? As well as for the ventilation impact, most things like viruses, things like mycoplasma, hang around much longer at lower temperatures. It increases shedding capacity of the animal and improves this viral stability, and it does, as I mentioned before, impact their mucociliary clearance. So actually temperature for calves is quite a big thing. Bedding is important, so clean, dry, and enough of it to try and it has been shown statistically to reduce the risk of pneumonia if they're kind of fully nested and bedded properly. So the kind of things we see, overstocking, no drainage would be the kind of big ones. Poor ventilation is there certainly in some sheds. Maybe it's something that we do look at. Draft is a big one, but I suppose a lot can be mitigated by the stocking rate. And as I mentioned, they're not treating kind of the dairy and suckler animal the same. So just the last couple of slides now really are about trying to bring together those risk factors, and we can assess them, but what can we do about them? So ideally, we need to try and reduce the number of bugs that calves are in contact with, and we need to increase their immune system's response to that. So how do we go about reducing infection pressure? It's about hygiene. That's of the calving pens, the calf pens, the feeding equipment, finding those cases early, treating them properly and quickly and for long enough, particularly for things like BRD. Temperature control, so are they bedded enough? Is there any way we can increase the temperature for those particularly young calves, under a month of age, whether that's maybe up their plane of nutrition or introduce other things, maybe like calf jackets or so on. Minimizing the group changes, looking at the timing of disbudding, etc. Is there anything we can do to improve the housing to try and really reduce infection pressure, whether that's increased drainage, reduce stocking rate, etc. age matching of groups, and again, management of those sick ones, feeding the youngest calf first. Those are all things that will feed into reducing infection pressure. And maximising their immunity really is about doing everything we can to try and promote and maximise the colostrum management. So it doesn't, we mentioned if we stuff them with colostrum and put them in loads of crypto, they're still going to get sick, but they won't die. But very often there will be marginal gains to be had around colostrum, whether in calves, in herds where they are experiencing calf health issues. The nutrition is an interesting one, and of course very controversial, whether we look at this in terms of economics and labour versus health and welfare, and it needs to be somewhere that looks at all of those things. But the plane of nutrition ultimately does have a major role in calves getting sick or not. Their routine, they love routine, so it's not just about kind of feeding, but kind of routine of management, and certainly making sure they're fed at the same time each day, etc. Making sure that they're treated, sick ones are treated quickly, and that if we're using a vaccination protocol, that it's done properly. So I guess in terms of the calf diarrhea outbreak, the pathogen is important because it might mean ancillary drugs, like for example, if it's crypto or coccidiosis. But regardless of that, the colostrum management protocol, the plane of nutrition, the hygiene, is there anything we can do to clean more often, increase the bedding, etc.? Is there anything we can do to break the kind of infection cycle? So is it possible to try and divert calves for the second half? If you're in the middle of a crypto outbreak or something like that, is there anything we can do to try and break that cycle, or at least reduce the level of contamination that the calves are coming into? That might change the housing, what we wear, our boots, our foot, our foot dips, all that kind of thing. Looking at how we manage the sick calves might actually change the mortality, and how quickly they respond to treatment and so on. Cleaning utensils is a big one. So can we do anything around that to improve it? And looking at the actual husbandry, is there foot dips? Is there people that are moving around kind of willy-nilly from the youngest calf and sick calves and so on? Disinfection protocols, those things sound kind of airy-fairy, but they're actually pretty important. And putting in a sensible vaccination protocol to kind of focus the mind on, of course, on the colostrum then as a sequelae to that. And of course, looking at housing risk factors. So when we talk about SOPs for cleaning of equipment, this is very detailed, but ultimately the point is that you don't put plastic milk bars or buckets straight under boiling water so that we don't go and bond the milk fat to the plastic, which can create biofilms and so on. And this is very detailed and talks about different degrees, but I think it's simplified really by saying that they're rinsed first and then they're washed with really hot water. That's really the big thing. All colostrum and milk feeding equipment needs to be very well cleaned with hot water, and ideally with some sort of sensible disinfectant. Shed cleaning, I suppose, at this stage of the year, probably a lot of farms are calving already, so it's not going to be practical, but this is something that is done ideally when the calves are not there. So steam is brilliant because it kills most things. But I suppose being clear that cleaning of sheds is the first step, and the disinfection with a crypto or cocci-active disinfectant is only done when the shed is already cleaned. Again, it's a bit late in the year probably for that at the moment. So just to bring together the kind of respiratory disease side of things, similar to the scour outbreak, is there anything we can do to look for marginal gains in colostrum management, the plane of nutrition, general hygiene, stock and rate, bedding management, and so on? How are they being treated? Are they being treated quickly enough with the right drug and for long enough? So working to develop that, because in reality the farm's not going to get the vet for every single case. But they need to know when they do need the vet, and they need to know what does that look like? What does that, maybe it's a certain temperature threshold and that kind of thing. Can we address any environmental issues? So if it's temperature is a problem, can we add more straw, calf jackets? Can we block draft? Look at stock boarding between pens, under gates, at gable walls, that kind of thing. Is there anything we can do to block drafts? So that might bringing in some straw bales, bale breakers, making sure there's no drafts under doors, that kind of thing. Is there anything we can do to improve the wind effect, which might mean retrofitting the inlet with Yorkshire boarding or something like that, increasing outlet, or potentially looking at the introduction of a positive pressure ventilation system? Is there anything we can do to improve drainage? Can we cut channels in the floor, add slope, etc.? Is there anything we can do in terms of cleaning? Reducing stocking rate, not sharing airspace with older animals. Tackling concurrent stressors is an important one. So obviously they have to be disbudded, but making sure that that doesn't coincide with vaccination or a group change or weaning, adding analgesia to reduce the effect of or impact of disbudding, that kind of thing. Making sure the vaccine plan is bespoke and appropriate for the farm. So, farms are often buying back, I guess that's maybe a sweeping statement, but making sure that those that are farms that are investing in the vaccines are doing it properly, giving it at the right time, etc. And looking at how they're weaned, because obviously, post-weaning growth checks is a key time for BRD. And there will be some pathogen-specific measures, if there's mycoplasma involved, that kind of thing. So this is just to the last couple of slides. I know I'm stuck for time just there. One of the boards that was presented to the farms there at the farmer audience at the CalfCare events, just trying to bring some of those key principles to summarize them. And also this one, which can be a little controversial, but I guess it's really about trying to reduce the impact of what is a very stressful event, which is disbudding and or castration. And really it's just saying that local anaesthetic needs to be given for disbudding regardless of age, and in addition NSAIDs would be useful. So to summarize, I think for BRD and scour, we do need to confirm the diagnosis and the etiological agent. Of course, that is important, particularly, is it crypto, that kind of thing. But regardless of whether it's scour or diagnosis, the key areas that are risk factors around colostrum management, hygiene, and plane of nutrition, how they're stocked, are largely similar regardless of the pathogen. So goals need to be achievable. So maybe it is just as simple as tweaks in the colostrum routine, milk the cows quicker after calving, get a fridge, make sure that they're checking quality, that they're checking passive transfer. Maybe that's as much as can be done this year, looking at changing more things in year on year. I think it's important to engage with the relevant people, because often the calf rearers are not necessarily the person who's looking for the manager or the owner or so on. So making sure that that is kind of communicated clearly is something, at least in our experience, has proven difficult to achieve. We can actually look at training of farm staff, and certainly the bigger units, it would be my experience that they're mad for that kind of thing. Again, a little bit late to this day of the year. But that calf management and review of calf performance is kind of built into the routine monitoring and following up. Did it work? So that's all I have there. I wasn't sure of the time when I started, so hopefully, Michelle, if you're there.
Yeah. Thanks, Catherine. That was brilliant. Sorry, the sun's coming in here now, but we have a few questions there if you have time to answer them.
I do anyway. Yeah, go on ahead.
That was brilliant, by the way. If only we could all have a bit of your brain and information that you keep in it. But where can you get those long guarded swabs was the first question for taking an
For the deep nasopharyngeal swabs, we use mare uterine swabs, so they're the same thing. Just being careful if the calves are under 50 kilos, so if it's like a Jersey breed or something like that, they can snap off, so just being careful of holding the head up and so on. But they're just mare uterine swabs, so
they should be from any wholesalers.
They're usually fairly widely accessible.
What for taking bloods for the total protein and passive transfer and that? What is the age limit
for total protein? Good question. So you can split hairs on this. 1 to 7 days, 1 to 10 days, I would suggest would be fine. So kind of from 24 hours up to about 10 days is usually fine. If we think about it, it's they start making their own obviously from about a week of age. So that's where it becomes the waters are a bit muddied. But as a screen, 1 to 10 days, certainly 1 to 7 days is fine.
Great. And could you comment on the approximate sensitivity, specificity of the SNAP test for scour pathogens, and are most of the kits similar to each other, or is there variances in accuracy?
I don't know the numbers off the top of my head. I have looked at this before. So I think where the issue comes is that, they are susceptible to being out of date and that kind of thing. So I guess making sure that we follow them as per the letter of the law. I suppose where we have had issues is occasionally where, it looks like crypto and that's not come up in the first screen and things like that. But so they work reasonably well. We use them all the time, so I'm a big fan. I suppose what I mean is that, if it turns into a big outbreak and it's maybe just not fitting, that you haven't enough diagnostic power to say, look, this is definitely crypto, I would say that's where, don't be afraid to send samples in for confirmatory diagnosis, whether, ideally for crypto it would be a slide, the Kenyan stain or an antigen test. So they work perfectly fine, and I'm a big fan of them. I don't know the sensitivity and the specificity off the top of my head, but I would say follow them to the letter of the law. That's kind of the only issue I've run into with them.
Great. Just a question on the housing there is the effect of putting a canopy over the back of the pen in the colder house. Would you think there's a benefit to this in creating a microenvironment for the calves?
So I think that's a double-edged sword, to be honest. And I don't think it'll be a yes or no for every, a one size fits every house. So they work really well. Calves seem to love them. You'll find them lying in underneath them, and they're all cozy and tucked up. So if it's a big calf house, very wide, so more than 12 meters, it's very difficult to kind of heat, and they're not using loads of straw. If there's any questions around the plane of nutrition, etc., then they do work well. And calves love them, and the proof is in the calf will seek them out. I suppose where we have had issues with them is, in the face of a big outbreak of pneumonia, it can create like a micro environment of poor air quality. So on balance, I would say they're more beneficial than not. But I don't think every farm would need them. So I would weigh that up as to what is the biggest, if I think temperature is the biggest thing, then I might do that.
I guess making them easy to move is kind of important, isn't it, for that?
Yeah,
for that
you can be underneath them.
So you mentioned there about increasing the feeding rates of calves, and I just wondered, what's the max? If they still only want to feed twice a day, what's the max you can give in one feed?
Well, the reality, I suppose, is that voluntary feed intake, I would say there's no right or wrong kind of answer to that in terms of that it's this arbitrary line, because it'll depend on the size of the calf, because voluntary feed intake will be a function of body weight. Ultimately, if a suckler calf, for example, will take easily 20% of their body weight per day, and usually between 5 to 10 small feeds. So that's what they'll do if they're allowed to do it by themselves. There is no problem feeding 20% of birth weight. And in reality, for our 30-kilo birth weight calves on a 6-litre milk intake, they're feeding 20% of birth weight by accident or design. So 3 litres is about the capacity. And again, it'll be genetic dependent. Like there are calves that will be fit to take more than that. If there's crossbreeding Jersey, they might take less than that. So it'll really depend on the farm. 3 litres is kind of a relatively safe bet in kind of one feed. But that's obviously going to be caveated with the farm. So there's no reason why. And again, this is a lot easier to do if you've automatic feeders. Somebody who's feeding on a milk bar, is probably not going to start feeding 3 times a day. And I'm not saying that, but it needs to be obviously what's kind of practical and achievable.
Okay. Where can you send BAL samples? Equine Centre no longer takes bovine samples. Thank you.
Well, we certainly UCD Lab do them. So it depends on, so that's for cytology. They do any cytology is the same for lungworm. The BALs need to get to the lab really quickly, so that's actually really important how quick they get there. For the PCRs and stuff, like most of the commercial labs, we'll be doing a respiratory kit. So you need 2 bottles if you do take a BAL. So you need to put a few mils in a plain sample in a plain top tube for for PCR or your respiratory panel, and you need a EDTA sample for cytology.
Okay. Just one final question. So you mentioned about the importance of harvesting colostrum and doing it hygienically and the timing of the cow when she calves and when she should be milked. After what time does that start to deteriorate?
The second she calves, and if she's running milk, it'll be before that. So colostrogenesis is going on, right up until she starts running milk or she calves, at which point she starts to transition. So, a cow that's milked 8 hours later after calving, that has very high quality colostrum, that will still probably be okay in terms of 22% on a Brix reading, but the quality declines basically, or it starts declining from colostrum to transition milk the second to the calve. So the sooner, and as I said, sooner if they're running milk before. So you'll get away with it, I suppose, if they're, it's very good quality colostrum. But the sooner they're milked after calving, the better the quality colostrum.
Okay, so no one's allowed to sleep during the night then.
I didn't say that. I'm a firm believer. If you don't have a problem, you don't need to fix it. I wouldn't be getting up to harvest colostrum if there's no calf health issues. But it is something that we would suggest if there is a calf issue, that they, particularly, we're in passive transfer, that they might look at trying to get that colostrum a bit quicker.
Okay, no, it's great because it is kind of forgotten about the attention to detail there about colostrum. So that was a really good point, and hopefully people will have a good idea what to do, or a reminder of what to do for an investigation and that now. And just like to thank you again.
There's an Irish study that looked at the factors affecting colostrum quality, and you always get asked, is it the protein in the dry cow diet, is it this, is it the minerals, and so on. And actually the biggest factor affecting quality that was found was the timing since calving that it was collected. So that's under Irish conditions even. So
yeah, very good. Super. I think that's everything, and thanks again, and sorry for dragging you back early from maternity leave, but it was great. We've got loads of positive feedback already, so we have recorded and will be sent to all those that registered, and thanks so much. It was great, and thanks to everyone for joining.
No worries.
Thank you.
Thanks a million. Take care.